Hello there. This is the YouTube Space Lab project sponsored by YouTube and Lenovo, consisting of a project devised by Sterling Schreiber II, Alex James Pike, and Adam Joseph Warner of Miss Milsatich's physics class at Firestone High School. Our project deals with aerobic and anaerobic respiration of bacteria. In essence, our project will differentiate between these two types of bacteria by exposing them to an oxygen-deprived environment. First, let me define the terms that we'll be using. Aerobic bacteria require oxygen to survive, hence the name aerobic. Anaerobic bacteria require no such thing, often requiring, but not always, nitrogen. For our experiment, we will expose both anaerobic and aerobic bacteria to an oxygen-deprived environment aboard the International Space Station. Now, this experiment will entail the use of a light high-beam microscope with up to 100 times magnetic zoom. Now, we'll use a total of 5 cultures of bacteria for this experiment: gram-positive bacillus, gram-positive and gram-negative cocci, stratococcus pneumonia, and E. coli. Now, these bacteria will first be tested under the microscope to verify that they are in fact living at the beginning of the experiment. And then they'll be placed in a microscope slide in an oxygen-deprived environment and left to sit for 2 hours. Now, one important thing to note whilst conducting the experiment is that to view bacteria, what's called a gram stain is necessary for this process. A gram stain is a simple dye used to make bacteria visible to the human eye under a microscope. The bacteria will be exposed for 2 hours. At the conclusion of 2 hours, the astronauts will then return to the testing chamber to examine whether bacteria has survived. If they survive, the conclusion can be drawn that they are anaerobic. If they have not survived, then they are aerobic. Our hypothesis is that the aerobic bacteria will shrivel and die in the oxygen-deprived environment, while the anaerobic bacteria will survive.